don't forget, I think I did send an announcement about this. So this is open and available. It's a homework assignment. And it will be open until Monday at 11 a.m. So this is to get you more prepared for the second lecture exam, which is not this Friday, but next Friday. And then the lab quiz or take-home assignment is going to start this Friday. It'll be available Friday at 1 and go through till next Wednesday at 11. Okay, so just make sure you're keeping all that straight and getting those things done. All right, so stems. We need to think about stems. We've already looked at roots. So now let's think about dicot versus monocot with the stem idea. So that's what you're seeing here. So actually, the one on the left, the blue one, is a dicot stem. Now, when I first learned this, that got me utterly confused because what have I been showing you for a dicot root? Does it have a ring configuration of vascular tissue? No. Dicot, I always think about a plus, 2 parts of the plus sign, dicot. Dicot roots have that plus configuration smack in the middle of the root. But now we see a dicot stem is a ring. And that thoroughly confused me until I was shown an image that I'm going to show you in a minute to explain how their root system morphed from that dicot vascular tissue arrangement in the root with that plus sign into this ring of vascular tissue. So no matter what you're looking at, if it's stem or root, you should key in on the vascular tissue, find the xylem and phloem. Usually xylem's the easiest, phloem's right nearby somewhere. And then you look at its pattern of arrangement with respect to, all the other vascular tissue around it, and then the other parts of the stem or root. So the dicot stem on the left, where do you see the vascular tissue? Is it smack in the middle of the section or toward the perimeter? You see that difference? It's toward the perimeter. So that's something you should key in on. So think like an architect. If you were designing a skyscraper building, you don't want all of your steel beams in the smack center of the building. You want to create a scaffolding around the perimeter for better stability. Same idea with plants. With the dicot stem, it makes more sense to put their strong xylem, particularly the xylem is pretty strong. It has nice rigidity, structural support. So put that around the perimeter of your stem. That will keep you a little bit more upright. It'll help buffet against, if you have buffeting winds or something coming along and knocking into you, having that strength on the perimeter is a good idea to keep you upright and stable. Okay, the picture on the right is a monocot stem. And here, what they've pretty much zoomed in on is vascular tissue. You're seeing the drawing of a bunch of vascular bundles. So each one of these little things we would call a vascular bundle. All right, and you can see it's pretty scattered throughout the stem tissue. So again, training your eye to look at the design of where is the vascular tissue, how is it designed, how does it look with respect to the rest of the plant. So if you work your way from the outside in, you'll have an epidermis layer, especially with a young dicot. It'll still be an epidermis rather than bark, for example. And then you'll have a cortex region, and then a ring of vascular tissue. And so you can see they're calling it vascular bundles, which is accurate. Those are bundles, but the overall design is a ring. So in between the xylem and the phloem of this image is where you'll find the vascular cambium. That's something else you need to think about as you're looking at these, especially under the microscope. And with the vascular cambium, we say it leads to secondary growth. Primary growth is when you germinate a seed in the soil and the seedling starts to emerge. That's primary growth. If you get the lengthening of the root, the shoot lengthening to come above the soil. Secondary growth is this idea of, okay, now we need to increase the diameter of the stem. And so the vascular cambium is going to have that job. And it's a ring of meristematic tissue in between the xylem and the phloem. So it is in this region, if you can pinpoint the xylem, kind of looks purpley red, and they're large, okay? And then you see these, with each bundle, you see the purpley red, and then you see this kind of delicate bluish swath of cells, that's the phloem. So you have to go in between those two. So I don't know if I can get my pointer to, or my, so in between, whoops, yeah. Can you kind of see what I'm doing there? Okay, so that's the vascular cambium, and it goes all the way around. So it's in between the xylem and the phloem. So it's meristematic tissue that will go through regular cell division. And what it does, the vascular, when one of the cells, the vascular cambium, decides to divide, it will produce two cells, but they'll have different destinies. One will be a xylem cell to the inside, one will be the phloem cell to the outside. And so each time the vascular cambium cells divide, that's their routine. So put one of the progeny cells will be xylem to the inside, and then the other one will be phloem to the outside. And if you keep doing that, that keeps lending to the increase in diameter of the stem. And then usually the smack in the middle, they often refer to that portion of the stem as the pith region. It's parenchyma cells, okay? It's just a bunch of parenchymas. Okay, so let's compare that to the monocot idea. Monocot, well, they don't have a ring of vascular bundles or tissue. They start with an epidermis on the outside, especially if it's young. Well, they won't really ever get bark. But then they have vascular bundles that are scattered throughout the whole stem tissue. And then we just say there's a bunch of parenchyma cells in between. They'll call it the cortex region. Okay? But again, the cortex region, when you look at it, it's a bunch of parenchyma cells mostly. And then the thing here is if you look, there really isn't any vascular cambium. There's no ring of meristematic tissue like what we just saw with the dicot stem. So the consequences of that are that the stem of monocots can never really get that big in diameter. They don't have an easy way to do that. And so monocots tend not to have, there's a couple of exceptions, but monocots tend to be fairly modest in their stem diameter. Okay. So looking at this is showing some vascular tissue through actually several groups of plants. They're talking up top about the seedless vascular plants. We really haven't gotten into them yet, so you'll see the slide later. But what I want you to look at is here, where you see this ring of tissue in the middle of the images here. And again, you can see a ring of tissue. It's just that they're more separated from each other, the vascular bundles. So the vascular bundles can be like that, where they'll be like distinctly separate from each other, but overall it's a ring, or they could be pretty solid ring. Okay, so there's variations, but in general, you see a ring of vascular tissue, and then the monocot example, it's scattered, all right? So we have to think about this dicot stem is a ring, but the dicot root was that plus configuration idea. in the center of the root. So this image really solved it for me. Once someone showed me this, like, oh, so down low here, you have the root tissue where they're trying to show that plus configuration of the xylem, and phloem is in the corners. And then there's a region of right between the root and the stem that they call the transition zone. And what you see is this gradual shifting into the circular ring of tissue. All right, and so if you were to take some very fine sections here and look at it in a microscope, you would see this gradual shift from the plus design to the ring design. All right, so they have solved that. It's not this huge abrupt shift. They have this nice gradual shift and keeping a nice connection all the way through. Okay, so it can happen. So now that we're getting into stems, you have to be able to tell apart, okay, what am I looking at, stem versus root? And then, okay, is it a dicot or monocot? So you have to start training your eye, which we're trying to get you to do both, looking at these, but also what you do in the lab exercises. So again, the dicot stem, you have that in this the vascular bundle, but they're pretty close to each other. And overall, it's a nice ring of tissue. And then the vascular cambium, xylem, and these cells over here are the phloem. It has fibers here. So you have fibers on either side of the vascular tissue that are just helping protect. Vascular tissue is pretty precious, so it makes sense to have that protection. Right? So ring of vascular tissue versus scattered vascular bundles, dicot to monocot. Let's zoom into one of those vascular bundles. So they often refer to it as a face-like design, where you can see the eyes, nose, and mouth are the vessels of the trachea, of the xylem. It's the vessel type of xylem. And then the cheeks and chin tend to be the tracheids. And then the forehead is usually the phloem tissue, sieve tube elements, and companion cells. So each one of these vascular bundles, if you were to zoom in, you might see something like this. Now this is, someone drew this. It's not an actual slide specimen. Often they don't look quite so distinct, like that's an actual slide of a monocot stem, and you can't really see the face, but this one is a little bit better. Okay. So it just, it depends on the plant species and how good, excuse me, how good the prep was. But it's very often that, we introduce the concept using an idealized drawing like this. Okay, vascular bundles, xylem and phloem together. Okay, let's go back to the dicot stem. So this is nice because they've color-coded it so that the pink is all the xylem, the red line is the vascular cambium, and then the green, just to the outside of it, is phloem. Now, the way the artist drew this is a little lopsided, right? Where, you see way more xylem, right? Compared to the phloem, even though I just told you a few minutes ago, every time the vascular cambium divides, it produces xylem to the left, or to the inside, phloem to the outside. So you would think there'd be even-steven amounts, right, of xylem and phloem. And that was another confusing thing when I was first learning this. Like, what happened to all the phloem? And what you have to realize is that a lot of the phloem dies off. And so you get really just a narrow band of living phloem. And especially when you think about dicot stems where you get a more mature, you're past the seedling stage, you're into bark tissue, you have a tree. What happens is with that bark kind of having a force, kind of keeping things from not getting too big in diameter, you have that pressure. And so as you get that growth outward, some of the older phloem will die off, but you still have a ring of living phloem. So that's why you get this imbalance, so to speak, of you get a lot of xylem and just a little bit of phloem, even though that vascular cambium is dividing to produce xylem phloem each time. Okay, another thing with this image, that xylem, the pink, you see how there's like this banding pattern in that region. Those are annual rings that are pretty much a history of that plant's life to date. And so this one, it looks like it has 1, 2, 3, 4 swaths of annual rings, so 4 years old, roughly. And that's another thing you can look at carefully, even just with minimal magnification. If you have a tree trunk and cross section, you can count the annual rings. And depending on the width of the annual ring, that tells you how much growth happened that season. If it's a really wide ring, that means it had lots of growth, which implies really good growing conditions. They had adequate water and their environment was good. Sometimes the annual rings are very narrow, which means they had a tough time growing that season. There's some variations. Different species tend to have different sizes of annual rings, so you have to be careful about that, but you can tell a lot by the annual rings. We'll look a little bit more at that. Okay, so that vascular cambium, it's a meristematic region of the plant, and it is leading to that secondary growth, which is increase in diameter. We have that xylem toward the inside and the phloem toward the outside. So all that xylem that's getting produced, technically that's wood. That's how you would define the word wood in a botany class. You would say it's secondary xylem that's getting laid down vascular cambium. And so again, with all those annual rings of xylem, it's usually the newer ones that are the most active, and then the older xylem tends to stop functioning as water transport. It's still good for structural support, but it doesn't, it usually gives up the job of water transport, and that is for the newer, more recent xylem. And remember, with xylem, that secondary cell wall is containing lignin, which is super strong and very good structural support. Then to the outside of the vascular cambium is that secondary phloem. And technically that's part of the bark tissue. So when we look at bark, which we will in this slide set, the definition of bark is everything to the outside of the vascular cambium, which includes phloem. So phloem is actually part of the bark. We'll look at that in more detail. And again, the more recent phloem is active, the older one actually dies off and disappears. You don't see it. So they break open from the pressure of the secondary xylem getting laid down, and then also with the bark to the outside. It's kind of squeezing them. And so the phloem, older phloem, dies off. So here's a nice cross section. So how many years of annual rings do you see here? How many? Just the 3. So the middle is different. So we don't count that. We count 1, 2. So 3 years. So that's a young, relatively young dicot. And then you can see the good shift in color and design between the xylem and the phloem. So then you have that ring here is the vascular cambium, and then the phloem to the outside. All right, so the phloem is in patches here. So here again, just trying to show the demarcation between the xylem and the phloem. So that ring of cells is the vascular cambium that will be dividing all summer long. Okay, let's try an eye clicker with this one. Let me see if I can get this to work. Okay. So just for the first question, just answer the first question. Which is a stem? Which is a root? First, just think about that. If you're trying to decide stem versus root, try to think like an architect. And where would you put your vascular tissue if you were below ground or above ground? Yes? So just choose either A or B. Oh, I'm sorry. Thank you. Which is a root? Sorry. In my head, I had it all straight. Which is the root? Answer that. Which is the root? A or B? Have we all chimed in? Okay. Can you see the bar graph? Yeah, it's freaking out on me again. So I asked which is the root, and 85% of you said A. That is correct. Okay. Why is A root tissue? Yep. It has the vascular tissue closer to the center. So if you're buried in the soil, you have all that support of the soil hugging you. You don't need to worry so much about having strong structural support to buffet you against, to help against wind, pressure, or someone bumping into you. Okay, so A is the root. Okay, let's do another clicker, and this time I'll be more clear, I hope. So now the next question is, looking at A or B, which one is a monocot? Which one is a monocot? Ten more seconds. Okay, looks like everybody has clicked in. So 89% of you said A. A is the monocot. You were correct. Okay, that's a monocot root. Monocot roots have a ring of tissue, but that ring of tissue, because it's root, it's closer to the center, not so much on the perimeter. B is stem tissue, because the vascular bundles are right on the perimeter. And it's a ring of tissue. This must be a dicot stem. Because a monocot stem has scattered vascular bundles. So I know this is a little, like, you got to think through it. It's a little confusing, but you need to be able to tell the difference between, am I looking at a root? Am I looking at a stem? Am I looking at a monocot versus a dicot? And so please use this as a study tool, and you'll do more of this in lab too. Tomorrow you'll look at a bunch of this. All right, let me see if I can get back without this freaking out on me. I don't think it's gonna happen. I'm actually going to come back. All right. Oh, let me just get back into sharing. Okay. So when you look up the definition of wood in a botanical sense, it's secondary xylem. That's what you're really thinking about. And it's looking at all the growth rings. If you have an older tree that's been cut down and you're looking at the cross section of the tree trunk, you'll have some definite growth rings. That's all xylem. And depending on the tree that's cut down, you'll see with angiosperms, you'll see spring wood versus summer wood. And so when they say that, so if you talk to a forester, they might use these types of terms. Spring wood. The spring wood will be larger cells in diameter. The summer wood will be smaller cells in diameter. So basically what they're pointing to is that the spring wood tends to be vessels. Remember, vessels are xylem that are larger in diameter. Tracheids are xylem that are smaller in diameter. So spring wood tends to be more vessels, and then as summer kicks in, you'll see more tracheids. And there's a definite reason typically for this. If you are a tree and in the spring, if there's enough in our region of the world, enough snow, melt off, lots of rain, that soil that you're growing in is going to have a good amount of water. And so to take advantage of that, you'll lay down a lot of vessels. Vessels are more efficient at water transport. So put down vessels, grow vessels, keep doing that until you start to sense, okay, spring is over. My water supply is a little bit, it's not so generous. So I better be a little careful here. I'm going to start laying down tracheids. My new growth will be tracheids, which aren't as efficient, but as we start learning about how water is moving to the xylem, putting tracheids down when you're a little bit like, yeah, I might go into drought here. Tracheids are your better bet. I'll explain why. Okay. And so you'll see this shift in diameter. You'll have vessels in the spring, tracheids throughout the summer. And so I'll show you a picture of that. And then heartwood is often referred to. That's the center, especially of older trees, where the center looks discolored, it's darker, it's pigmented darker. And that's what they often refer to as heartwood. That's where the tree has laid down quite a bit of resins or tannins or gums. That these are secondary metabolites. These are chemicals that the plant is making, the tree is making, in order to kind of protect themselves in this region against fungal attack primarily, but microbial attack in particular, so that that portion of their body is not going to get attacked. And then sapwood is a term that's often used. Those are the lighter areas, the active areas of the tree trunk, where you're going to have really active xylem, active phloem. Okay, and so you'll see the sapwood idea. And so these terms often get thrown around. I wanted to define those a little bit so we're kind of clear on what that is. So here, this is nicely labeled in terms of the xylem and phloem. They're referring to this as epidermis, so this is very young. This isn't a tree that has bark yet. Here's a really nice distinction between vessels and tracheids. So the summer, spring, a lot of vessels, and then summer, a lot of tracheids. So this is a long growth, good growth here. Lots of tracheids, still good growth. And then the next season, spring, you see this spot is narrower, so their summer here was not maybe they had more drought, some other issues going on that caused them to slow down. Okay, so vessels and tracheids. Now, if you get out of the angiosperm realm and go into the gymnosperms, so pine trees are gymnosperms. They do not have vessels, remember. So now if you cut down a gymnosperm, a pine tree, and look at it in cross section, you're not going to see that lovely pattern that we just looked at. You won't see that. So growth rings can be a little bit more challenging, but they're there. And, you know, they can kind of widen their tracheids somewhat, but it's not nearly as dramatic. So you can see here, this cross section, the xylem looks pretty similar in diameter all the way across. It's certainly nothing like that. Okay, like that image. So again, just remember that gymnosperms don't have those vessels. And so you have a more even look to the diameter when you look at the xylem. And then here you can clearly see heartwood versus sapwood. And again, the darkening is just the trees, the plant's ability to try to protect itself by these secondary chemicals that get put down that are discouraging any infiltration by any kind of pathogen. Yes. Yeah, so here, this is more for the center of the tree. Still have some xylem, some vascular. So they, this probably, I don't know what species this is, but that dark area is the kind of non-functional xylem because it's been kind of impregnated with all the different secondary metabolites. So the lighter part is the active xylem, and then the vascular. So with hardwoods, sometimes you'll hear this designation too, hardwoods versus softwoods. So if someone's talking that way, generally what they mean is a hardwood is a dicot tree and a softwood is a gymnosperm. So often you'll hear people talk about quality of wood. And when I was a grad student up at Michigan Tech University, I used to rent a huge house full of grad students, and most of the grad students were foresters. And so we would often have all kinds of conversations where they throw around these terms over coffee in the morning. So it is, you know, things you hear a lot if you are into wood making or, you know, want to go buy some lumber. So something to think about. Okay, so what is bark? So here we need to think about, okay, once the tree or plant is starting to get more mature, they will start to shift out of having just the epidermis, and they're going to start to get into manufacturing bark. And of course, bark is super protective, and its overall design is very nice to protect. And by definition, bark is all the tissue outside the vascular cambium, including the phloem. And so everything outside of the vascular cambium. So we often refer to that phloem as secondary phloem, just like we talk about the xylem as secondary xylem. Okay, it's being formed by the vascular cambium. So technically, yes, it's secondary phloem. And we already talked about the periderm back when we were looking at stone tissues. So, you know, initially all plants have an epidermis, and then some plants will shift and start forming the so-called periderm. And then over time, the bark. All right. And so it's just this process that they're going to go through. And so in order to get the periderm and bark to form, you have to have cork cambium. And so this is another ring of vascular tissue. So when you look at a more mature tree, they'll have a vascular cambium that's giving rise to xylem to the inside, phloem to the outside. And then further out toward the perimeter, they'll have a cork cambium. And the cork cambium, its job is to make the bark tissue. And so, except for the phloem. And so here, the cork cambium is going to divide and it's going to put, let's stick with the rhythm here. It's going to put parenchyma cells to the inside, which we often call cortex cells, but they're just secondary parenchyma cells or cortex cells to the inside, and then cork cells to the outside. So very different cell types, because the secondary cortex cells to the inside stay alive. The parenchyma cells will stay alive for the most part. And then those cork cells that are being produced to the outside, once they become mature, they're going to die. But they'll stay there. Okay. And so they get to their full size, they get the signal to go ahead and die off. But before they die off, they'll get impregnated, the cell wall will get impregnated with suberin wax. So suberin is a type of wax. And again, it makes sense that the bark, outermost layer of the stem, the bark, is going to have some water retaining properties. And so the wax in the cell walls will do that. So bark is the cork cambium. That's, the cork cambium is really doing the bulk of the bark formation. The only thing it really doesn't make is the secondary phloem. All right, so the vascular cambium does that, and then the cork cambium does everything else, the bark. And bark can be incredibly diverse and sometimes really hard, like with a naked eye. Sometimes you'll see lenticels. It can be very smooth, it can be very rough, it can be peeling away. And so there's huge diversity in bark overall. And I think just from you living your lives, you've noticed that over time. Look at that bark. Very, very beautiful. So that's not photoshopped, that's real. And unfortunately, we don't have that here. Wouldn't that be awesome against the white snow? That would look so awesome. But you have to go well south. So I guess they're native to south of the equator. I think Hawaii has some good patches of them. Has anyone seen these? These are called rainbow eucalyptus. You've seen them? Cool. Was it that vibrant in Hawaii? Yeah. I have a friend that goes to Hawaii frequently, and she's taken numerous pictures of those. I've never been, but and then what happens is this will peel, and so if you have that tree, it will look different over the season and next year, because the bark will peel and reveal different colors, and so it's like really cool because it's always kind of slightly changing, giving you a different look. I think we should try growing this in the greenhouse. Don't you think? I wonder if it would respond this way and keep that kind of growth pattern. It's Eucalyptus deglupta. That's the scientific name of the rainbow. The common name is rainbow eucalyptus. So beautiful. Yeah, that would be awesome. Bonsai you could have in your house. So, and then you go to this kind of bark, very, very not like what we just saw. And again, it can get quite thick depending on the tree species. The bark can be very, very thick. It can be very thin and papery. By the way, going back to like this middle image of the birch. So we have birch trees around here, and you know how it kind of peels off and you have like dangling bits. And when I would go hiking with my boys when they were toddlers, that dangling bit, especially if the wind is flapping it, it's so tempting to take it and just keep peeling. Don't do that. I'd have to yell out to my boys, stop doing that, because if you take that and essentially peel the bark all the way around in a circular pattern right there, what you've done is you've girdled the tree. You've removed the phloem. Remember, phloem is part of the bark. And so if that peel is deep enough and you do it all the way around, you're girdling the tree. You've killed it. And now it's going to go through a very slow death because it is, for a while, the tree's going to act normal and behave normal. But what's happening is, especially with photosynthesis and all the sugar production, then the leaves start sending the sugar down. It's like if this is quite a girdle, the sugar and the foam will come to here, but then nothing, no more foam here. And so it just starts to build up, and they can actually show that this part of the stem is well because there's so much sugar here, but it can't continue down to the roots. So then the roots starve of sugar. They don't get anything, and then they die, and then the rest of the tree dies. So it's a very slow process.
Like, let's say we cut a oak tree in half, because my dad's done that, and for some reason turned into a book. It refuses to give up.
Yeah, with oak trees, is it creating suckers? Is that what it's doing, like from the base of the stem or the roots? Yeah, yeah, yeah. I think sometimes it can depend on the time of the year that happens. Both of those situations. So yeah, you got to be, and I think at least in ecology classes, they probably do they talk about that patch of birch trees out west? There's a name for it. It's, of course I can't remember, but it's just a giant clone of birch trees. There's a name for it, and it's famous. It's been well studied, and now they're saying it's dying. Pando, thank you, pando. I knew it was a unique word. And so they're worried about it because it seems like the patch is starting to die off. And so they've been studying it long term. But birch are famous for, if you see a cluster of birch, they're probably clones of each other. And it's probably because they're sending up new shoots every spring. And it's just a massive clone. And so it's always fun to come across that kind of growth pattern in the woods. So here is a little bit more on bark in detail. So up top is a younger bark where you have the cork cambium, the dark brown. And so when it divides, it produces, you don't need to know the word cellular, by the way. About that. I would want you to know that when the cork cambium divides, it's producing parenchyma to the inside. They're living. I would like you to know that. Ignore cellular, I'm not kidding. And then the cork cambium, when it divides to the outside, produces itself to the outside, that's a cork. So that I want you to know. So the upper image is younger. The lower image is a more mature plant where you can see layers of cork building up to create a thicker bark. So over time, you can get a thicker bark depending on the plant species. So here is an image under the microscope where the cork cambium is, you can kind of see the difference in the pattern of the cells. Excuse me. So the cork cambium is in this region here. Whoops. And the more kind of brick-like cells are the cork. So you have a big swath of cork here. That's a big, big section of cork. And then the living parenchyma cells are to the inside. They're more roundish. And then this one's nicely labeled. So these are older terms, phloem, phloderm. I don't care that you know those. They're older terms. They're outdated. Cork phloem. So cork cells, the cork phloem. Yeah. So here is the cork cambium, this line of cells. And so the cork phloem are cork cells. And then to the inside here, those are living parenchyma cells. Okay. So I have in the notes section the definitions too. Here's a nice image showing both. So let's look at this a little closer. So they took a swath of this section and enlarged it. So you can see on here both the vascular cambium here. There's the vascular cambium. And the vascular cambium's job is to give rise to the xylem to the inside and the phloem to the outside. And then further out toward the perimeter, you have the cork cambium. Right, you can see it's way out there. And so the cork cambium is busy doing parenchyma cells to the inside and cork cells. So those two rings of tissue are very important for increasing the diameter, laying down new xylem and phloem, which increases the diameter. And then the cork cambium's main job is, for bark, most of the bark. So then the bark is pretty heavily suberized, got that wax, but the living parenchyma cells just underneath, as well as the phloem and, the cells near where you see all the secondary phloem, a lot, those are living cells. So you still have to have oxygen in that area. And so the bark lenticels will help with that. So the bark lenticels we talked about already in cells and tissue section, but again, it's this more loosely designed patch of cells that allows for better airflow and access to oxygen. Because those living cortex cells, the parenchyma, the phloem, all of those cells have to stay alive. They need oxygen to do that. And so the cork cambium's job is to also make sure that there's some regular lenticels in the overall design. And sometimes you can see them with the naked eye like that, but sometimes you look at bark and you're like, where are the lenticels? They're there. You just can't see them because of the roughness of the bark. And then some apples and pears will have lenticels, but they're not like produced from a cork cambium, but they call them lenticels because it's the same function. It's these looser patch of cells to allow for airflow, essentially. So not because of a cork cambium per se, though, but it has the same function. So bark lenticels are helpful. Here's some that you can see with the naked eye. Here's a real obvious, with the naked eye, that those large bumps, those are all lenticels. And then under the microscope, you would see that bump showing up on the edge. Whereas, again, if you were to describe that, you say, well, it looks like they're loosely packed, kind of scattered. So that's a good area for airflow to come across. And then some lenticels will look a little different, like this is birch. And so their lenticels are a little bit different. But okay. So as you're working through in lab tomorrow, you'll be training your eye to what we just covered today, looking at stem sections, understanding that it's a stem, not a root. Why is it a stem and not a root? Keying in on the vascular tissue, looking at lenticels. And then tomorrow you'll also do a lot of live prep. We'll have a lot of living plants that we have bought for you that you will be sectioning and looking at under the microscope. So it's a fun lab tomorrow. All right, we'll stop there. We'll finish off stems on Friday and then get into leaves.